Good morning, everyone. So today I am with another topic, and the name of this topic is control and coordination and homeostasis. From the current topic, from the current syllabus, that is IGCSE Cambridge program, and the topic is chapter 14. So before going into the detail of the topic, let me show you the syllabus guideline. So as per the syllabus, we have to do coordination and response, that is 14.1. And in this section, we'll talk about electrical impulses, how they travel. Then we'll describe the mammalian neural system in terms of central nervous system and peripheral nervous system. Then we'll describe the role of nervous system as coordination and regulation center for the body. Then we'll see the diagram where we'll see the images of sensory relay and the motor neuron. Cambridge asks question from here. Sometimes they ask to label the cell diagram or to discuss about the synapse. So they can ask in which direction the impulse is flowing. So you have to put the arrow. Then describe a simple reflex in terms of receptor, sensory neuron, relay neuron, motor neuron, and effector. Describe a reflex action. Then describe synapse. Then we have to describe the structure of synapse and describe the event also. So in the event we are just going to discuss how impulse travel. So we'll talk about impulse stimulate the release of chemical neurotransmitters into the gap. Then neurotransmitter molecule diffuses across the gap. It binds to the receptor molecule on the next neuron, and in this way impulse is generated in the next neuron. State that synapse ensures that impulse traveling in one direction only. Then we have 14.2, which is sense organ. In this, we'll discuss about the receptor cells, such as the light receiving cells in your body, sound receiving, touch, temperature, and chemicals. And in the diagram of, so as the topic is sense organ, in this topic, we'll just discuss about the structure of eye, limited to cornea, iris, pupil, lens, retina, optic nerve, and blind spot. And then we'll discuss about the function of these parts. Explain the pupil reflex limited to change in light intensity and the pupil diameter. Explain the pupil reflex in terms of antagonist action. When we say antagonist action, that means they are a pair of muscles work in an opposite way, like circular and radial muscles. They are antagonist muscles. So one is going to relax, and when one is going to relax, at that time another will get contract. So we'll see in the example. Then we'll talk about accommodation, describe the distribution of rod and cone cells, outline the function of the rod and cone cells limited to sensitivity of the rod for night vision. And there is a three kind of cones absorbing light of different colors for color vision. Then identify in diagram and images the position of fovea and its function. Fovea is a depression on the retina. So we'll discuss the function also. 14.3 discuss about the hormones. So first we'll discuss about what is hormone. Then identify in diagram. And in this diagram we'll discuss about the endocrine glands limited to adrenal gland, pancreas, testis, ovaries. Then describe adrenaline as a hormone secreted in fight or flight situations. Effect will be limited to increasing breathing rate, increasing heart rate, and pupil diameter. Then compare neural and nervous and hormonal control. Again, limited to speed of action and duration of the effect. State that glycogen is secreted by the pancreas and describe the role of adrenaline, control of metabolic activity, limited to blood sugar concentration that is going to increase, and increasing heart rate. Homeostasis is the last subsection of the topic. In this section, we'll talk about homeostasis as a maintenance of a constant internal environment. State that insulin decreases blood glucose concentration. Explain the concept of homeostatic control of negative feedback with reference to a set point. Describe the control of blood sugar concentration by the liver and the role of insulin and glucagon. Then we'll talk about type 1 diabetes. Identify and diagram and images of the skin, which is limited to hair erector muscles, sweat gland, receptors, sensory neuron, blood vessels, and fat tissue. Describe and maintenance of a constant internal environment in mammal in terms of insulation, sweating, shivering, and the role of the brain. And then we'll discuss about the maintenance of a constant internal body temperature in mammal in terms of vasodilation and vasoconstriction of the arterioles. And the last is tropic response, in which we discuss about the plants. So describe geotropism, then describe phototropism. Investigation is also there in terms of both. Explain the role of auxin in controlling shoot growth. So we'll discuss about the hormone auxin here in this topic. So as we have seen, it's quite long and very important in terms of exams also. So without delaying, let's go to the topic. Okay, so let's continue. So this topic is interesting because we are going to discuss about the brain, about the hormones, and how the body coordinates with all these things. And this topic is also important in terms of exams. So let's go ahead. So let's talk about the mammalian nervous system. So we have a two main neural system, the nervous system inside your body. That is central nervous system called CNS and peripheral nervous system PNS, right? So central nervous system is actually consists of two parts. One is the brain, and brain has extension which goes down as a backbone, that's spinal cord. Apart from this, all the nerves which is present inside your body, which is connected brain to other parts and spinal cord to the other part, they belongs to peripheral nervous system. So except brain nerves and spinal cord nerves, whatever nerves are spread throughout the body, they comes under peripheral nervous system. And what they allow, what is the function of these? Make sense of our surrounding and respond to them. And at the same time, they will coordinate and regulate the body function. So like if something is going outside the body, like suppose there is a rain or there is a sudden temperature change or there is a cold climate or hot climate. So body senses all these changes which is going nearby and regulate the internal function in such a way that it should not disturb the internal function of the body. So whether it's a hot climate or cold climate, your body temperature will be as it is. It's not going to change, right? And while controlling and regulating all this, information is sent through the nervous system in the form of a nerve impulse, and these impulses are in the form of electrical signal, right? And who is passing these electric signals? We call them neurons, and the bundle of the neurons is known as nerve. So that's when we say nerves, the meaning is there are so many neuron cells. So here we go. So this is your nervous system. You can see brain and spinal cord, which is a central nervous system, and the nerves which is present inside your body. They comes under and if we can see the one nerve, right? See this shows the one nerve is actually one nerve is a bundle of neurons, and you can check one. So this is one neuron cells they have shown here. So this neuron cells is a unique structure. Why unique? Because they are the longest cell. They are differentiated cells. Differentiated means they perform only one function, and the function is to control the transmission of the nerve impulse, right? So these are the cell body. Cell body receives the information and travel down through this tube to the axon terminal. This is axon and this is axon terminal, and another neuron is also connected to it, and in this way, not connected, they are near to each other, so that information will transfer from one cell to another cell. Okay, so let's talk about what is neuron. So neuron is a long fiber, right? It's a very long cell. Take less time is to transfer the impulse from one cell to another. So they are highly active cell. The axon is insulated by the myelin sheath, which is a small insulated area on the axon with some uninsulated area that's called nodes of Ranvier. This means that the electrical impulse does not travel down whole axon, but it's going to jump from one node of Ranvier to another node of Ranvier. So you can see here if impulse is here, so they're going to jump like this. You can see the cursor. And then again from here to here, so here to here, right? So like this. So the impulse are jumping. So it makes the transportation faster if impulse is jumping like this, right? Now the question is how body receives this information. So the cell body of the neuron contain some processes coming out from the cell body that's called dendrites. This means they cannot connect to many other neurons and receives impulse from them, forming a network of easy and fastest communication. Here we go. Let's see the structure of the neuron cells. The first one, you can see this one where my cursor is. This is sensory neuron, and in case of sensory neuron, the cell body is in the middle or somewhere near to the terminal, right? And one axon terminal is connected to the receptor cells. Another axon terminals may be connected to neuron or connected to the nerves to other cells or to the brain or the spinal cord. Relay neuron is the shortest neuron because they have just a cell body and a very small axons extension. Generally, in some cases, axons are not there. There's just cell bodies there, right? And in motor neuron, you can see this is a very common structure of the neuron cells we generally draw. So you have to understand this whole diagram. So let's go in the detail. So this front part on the top where my cursor is surrounding the structure, that's top called cell body. Inside the cell body, this is the nucleus, this is mitochondria. These are the beads are ribosome present on ER. Cytoplasm has extension making a long tube, right, like this. This is called axon. Axon is covered by the fat layer called Schwann cell, also called myelin sheath, and there is a gap between the fat deposition, that gap called node of Ranvier, and this whole long stretch is called axon. So we got to know three different types of neurons, right, in the previous slide. So sensory neuron, as the name suggests, sensory. That means they are going to carry the impulse from sense organ to the central nervous system, that is brain or spinal cord. Then let's talk about the motor neuron. So what motor is doing? They carry impulse from central nervous system to the muscle cells, effector organ, right? So always remember, sense organs are the receiver, muscles are responder. You respond, right? Then now we have to connect the sensory neuron and the motor neuron. So for connecting, we have a relay neuron, and they are generally found inside the central nervous system, right? So sensory neurons are long and have a cell body branching off the middle of the axon, whereas the relay neurons are short and a small cell body at one end with many dendrites branching off. So there are so many processes comes out. Motor neurons are long and have a large cell body at one end with long dendrite branching off it. So we have seen the diagram. See, it defines the same thing. So yes, this is the flow, how the information is flowing. So let's go through it. So this is the receptor molecule, right, on your skin, like sense organ, right? Your eyes. Information from the receptor molecule goes to central nervous system through sensory neuron, and the sensory neuron sends information to central nervous system. Central nervous system will receive the response, and they will do some appropriate um, information to the muscle cells, right? So they will send information to the motor neuron, and the sensory neuron and the motor neuron inside the central nervous system is connected by relay neuron. And once motor neuron will got the response, what they have to do, they will show the effect. Right? Effect on the body, right? Or muscles, like take an example how this whole process goes on. Suppose you are touching a hot object, right? So the plate is hot, that response goes to brain sudden gives an information it's hot, we have to leave, right? And if you're going to wait for 1 more milliseconds, then brain will give one more information, like it's made up of glass, if you're going to leave it, it will break, so you can keep it aside, right? So that informatory information is also coming from the brain through motor neuron. That's why your muscles works and you keep the plate aside. You are not going to lift it. You are not going to leave it, right? So it will. So in this way, the information travels down in the body. Reflex arc. Wow. So reflex arc is the total pathway. On the right side, you can see I have made a sequential pathway of the reflex action, right? So first, in the reflex arc, your first stimulus, which is going to receive is your receptor molecule, and receptor molecule sends information to sensory neuron. Sensory neuron sends information to the relay neuron, and the relay neuron sends information to the motor neuron, and motor neuron sends information to effector organ, and effector will do the response, right? So in this case, receptor molecules are maybe we have different types of receptors, right? Such as your touch receptor in skin, sound receptor in ears, light receptors in eyes, chemical and taste receptor in nose, tongue, and mouth, temperature receptor in skin and brain, right? So these are the receptor. When it comes to effector molecule, which is going to respond, is always muscles or gland, right? So gland also secrete hormone, right? So in one line, if I have to say what is reflex arc, so you can say reflex arc is the pathway through which body brings reflex action. And what is reflex action? Reflex action is a fast, automatic, and protective response of a body. So body gives all these things, right? How we can experience the reflex action? So in the diagram you can see there is a pin, right? The pin is detected by a pain receptor, or you can say touch receptor, or the pressure receptor in the skin, right? And once body will feel that, the sensory neuron sends electrical impulse, right, to the spinal cord, because brain is slightly away, right? So spinal cord is there. Next, after this, electrical impulse is passing to the relay neuron, and the relay neuron that is present in the spinal cord. Then relay neuron connects to the motor neuron, the green one, and passes the impulse on to motor neuron. And motor neuron carries the impulse to the muscles in the leg. That's why you pull your leg, right? So the pulling is the appropriate response. In this diagram, you can see one arrow is going upward and there's a star mark. The meaning is impulse is also sent to the brain, but the brain is not involved in coordinating the response. So just like an idea has gone, like somewhat happened to the body, that information has been reached to the cells, to the body, to the brain, right? Okay, so we all experience reflex action, in which this is the very basic example. Synapse. Now, as you can see in this particular animation, that this synapse is not a connection. Actually, it is a, you can say it's cells which are present in a close proximity. You can see there is no connection, right? So both cells are present in a close junction. So synapse we can define as a two neuron meet or join. They do so at a junction called synapse. So, and the gap between the synapse is called synaptic cleft, right? You can see here. So the basic diagram of the synapse is, this is the postsynaptic neuron, and the membrane is called presynaptic neuron, and the membrane is called presynaptic membrane. Inside we have a vesicles. You can see there are so many vesicles, and the vesicles are having neurotransmitters, right? Come to the next neuron. This is postsynaptic neuron, and this is called postsynaptic membrane. And on this membrane, we have a receptor molecule, right? So see, so the process of the nerve impulse is very easy. How impulse is moving. Let's try to understand the 6 points. So this is neural one, the point one, an impulse arrive at the end of the presynaptic neuron. So impulse has been reached here. Now what is happening? the vesicles which are present in the presynaptic neuron, they are going and fusing to the membrane, right? Membrane of the presynaptic neuron. And in the third diagram, we can see once the vesicles get fused with the membrane, they are releasing the neurotransmitter, the chemical, into the gap, right? And on the next diagram, you can see that the neurotransmitters has been received by the receptor molecule on the postsynaptic neuron, and the neurotransmitters are going to trigger an impulse in the postsynaptic neuron, and in this way the information is going through. It's very important to degenerate these rest of the unused neurotransmitters, because if they are not going to destroy, they will keep on sending impulse to the next neuron that is not required, right? So these are must be degenerated. So you can see these are the vesicles, right, released into the gap, and then these are the receptor molecule. Now let's talk about the sense organ. So we have 5 types of sense organ inside your body. So first is the skin, and they are sensitive to pressure, heat and cold, and pain also, right? And how the sense touch and temperature. Second sense organ, we have tongue. They are sensitive to the chemicals, what we eat. So they sense the taste of the food. Nose, they are sensitive to chemicals and they sense smell. Ears, sensitive to sound and movement, sense to balance and hearing. Eye, sensitive to light, help in sight. Sense organ as receptor. So receptor are the group of specialized cell. So when we say specialized cell, the meaning is they are doing one function. And what is the function? To detect a change in the environment and stimulate electrical impulse and response. Right? So sense organ contain a group of receptors that responds to specific stimuli, right? And once the receptor cells in the sense organ has been stimulated, they start generating an electrical impulse, which is passed on to the sensory neuron, which carries the impulse to the central nervous system. And here, the response will be decided on, and the impulse will be passed to the motor neuron via relay neuron, of course. Without the relay neuron, you can't connect the sensory information to the motor response. So yes, relay will be there. The motor neuron carries the impulse to the effector, and effector carry out the response. Like you have seen something unusual, so you start running, right? You are frightened. Okay, so next diagram is the eye structure and the function. So let's start from the layer. So this purple color layer, the top layer is called cornea. Second layer, the red layer, orange layer actually. This is called sclera, right? The third layer, the blue layer, right? This is your ciliary muscles layer. Fourth layer is retina. Retina is the innermost layer. After that, there are the liquid present inside. Then this is pupil, this is eye lens, this is ciliary muscles, this is iris, right? This is fovea, retina, and sclera. They also have optic nerves. So optic nerve have function to receive the information, and whatever we are seeing, send it to the brain, right? Let's talk about the structure and the function. Cornea, as I said, let me show you the location. Here is the cornea, the top layer, right? So it's a transparent lens that refract light as it enters the eye. So they are actually helping in bending. Then we have iris. They control how much light enters the pupil. Lens, it's a transparent disc that can change shape to focus light onto the retina. What is retina? Retina contains light receptor molecule, and we have a two type of light receiving molecule, rod cells and cone cells. Whereas rod cells have function to detect the light intensity and the cone cells detect the color. Optic nerve. Optic nerve are the bundle of the nerves, right? They carry this impulse between eyes and brain. Pupil. Pupil is a hole that allows light to enter the eye. So the amount of light going inside your eyes is going to control by the pupil. Blind spot. Okay, this is very important. Let me go and let me show you where is the blind spot. Blind spot is the area on the retina from where uh, optic nerve will come, right? They are going out, right? So if at this point, this blind spot point, if the light is going to fall, you can't see that object because the optic nerve joins the retina. There is no light sensitive cells there. So of course, nothing will be sensed. Light falling onto the part of the retina will not result in any image being detected because the brain fills in from surrounding light. So we don't see a black hole where no light has fallen, right? So this causes a blind spot where we cannot detect an object in our peripheral version, even if it is there, right? Very interesting. Pupil reflex. So let's see how the pupil reflex occurs. So in dim light and in bright light. So in bright light, of course, we don't want that much light to get inside your eye because they will damage your retina. And in dim light, of course, we need to see something. So the size of the eye will be slightly bigger. So in dim light, the pupil dilates, means it's going to get widen up in order to allow as much light into the eye as possible. Even if it's going more light, it's okay. But in bright light, the pupil constrict in this case, and they will narrow down. And making the uh, area narrow down is a plus point because this will prevent more light to enter and cause damage to the cell. In dim light, the pupil dilates and this will allow more light to enter the eye to improve vision. Whereas in bright light, the pupil constrict, always remember, they get smaller because to allow less light to enter the eye to protect the retina from damage, right? So let's talk about the pupil reflex and which is coordinated by antagonist muscle action. So here we have radial muscle and circular muscles in the iris, which is the best example for antagonist pair. So in this diagram, in the dim light, as we discussed in the previous slide, that in dim light you need to see something, right? That is why your eyes, your pupil will get open wide so that more light will enter and then you can able to see. So diagram showing the eyes in the dark environment. So photoreceptor detects change in the environment, that is photoreceptor cells, which is present on the retina, right? That is dark. So when there is a dark or less light, radial muscles contract, circular muscles relaxes, pupil dilates and more light will enter the eye. In this way you will able to see. But what if there is a bright light? We don't need that much light. So the photoreceptor again detect the change in the environment, that is a bright light. So radial muscles will relax, circular muscles contract, and pupil will constrict. They'll become narrower, and in this way the less light will go into the eye. So you can see in these two conditions, these three things, radial muscles, here it is contracting, here it is relaxing. Circular muscles is going to relax here, contract there. Pupil dilates, get widen up here, pupil get constricted. So there are some muscles which are working in opposite direction. So let's have a summary. In dark light or in night sight, radial muscles contract, circular muscles are relaxed, pupil size will get widen up, and more light will enter inside the eye. But in case of bright light, it's already bright. We don't need that much light. So your radial muscles will relax, circular muscles contract, pupil size will get narrower, and less light will enter the eye. Accommodation means a fine focusing, right, where the lens bring about a fine focus within themselves, right? And these cells are actually elastic and its shape can be changed when suspensory ligament attached to it become tight or loosen up, right? So in the structure of eye, I have discussed about how they are attached. Now the change are brought about by the contraction of or relaxation of the ciliary muscles. When an object is close up, let's see what happens when you are seeing a near object. So always remember three things. Whenever you are seeing a near object, a close-up picture, ciliary muscles contract, and this will cause suspensory ligament to get loosened up. And in this way, it stops suspensory ligament from pulling the lens, which allow lens to become fatter, right? So they will become fat and light is reflected more. Reverse in case of when you are seeing a far object. In this case, ciliary muscles relax, suspensory ligament is tightened up, and the suspensory ligament pull on the lens, causing it to become thinner. So there's a big difference between these two different types of light. So summary is, in object far away, the light is reflected and the object is close by, or light is more refracted. So in this case, ciliary muscles is relaxed, and in refracted case, it is more contracted. Suspensory ligament is tightly pulled, it's slacked here. Lens is thinner, lens is thicker or fatter. So like this, you can see this is the image of the lens, right? How they are in the distant object. This is showing the front view, from the front how the eye will look like. And this is nearby when what happens to the eye lens when you see the near object. Rod and cone cells. These are the magic cells which detects light, and the rod cells is the sensitive to dim light, whereas the cone cells are related to giving different colors. So there is three type of cone cells which are sensitive to the different colors like red, blue, and green. And the fovea is the area on the retina where almost all the cone cells are found, right? So rod cells are found all over the retina other than the area where the optic nerve attached to the retina. There is no light sensitive cell at all in this area. So that is why we get the blind spot name. How the cells are differentiated, how the cells are functioning, what is their distribution, and what the comment about it. Rod cells sensitive to low light intensity and they detect shades of gray. They are distributed throughout the retina but none in the center of the fovea or the blind spot. And yes, they provide night vision for sure. You can recognize the shape but not the color because there is no light, and your rod cells are low light sensitive cells. Cone cells sensitive to bright light. right? And they are concentrated in the fovea region, also called yellow spot. There are three types sensitive to the red, green, and blue light. Donate eye. Let's give a big gift to the person who is really in need. Comparison of the neural and nervous and hormonal control in terms of how they are made up of. So nervous system made up of nerves, brain and spinal cord, whereas endocrine system is made up of glands. Type of message: neural system, nervous system carries electrical impulse, whereas endocrine gland controls the chemical hormone. Speed of transmission, of course, the nervous system will be very fast and the endocrine system will be slower. Length of the effect, short, is very short. Longer until hormone is broken down. Hormones in the human. So a hormone is a chemical substance that is produced by the gland and it is carried by the blood. And this hormone is going to alter the activity of either one reaction or more than one reaction. Right? So they have a chemical information from one part of the organism to another and bring about a change, right? So like suppose insulin started producing, started converting glucose into glycogen. So we have to stop this conversion at some time, right? That is very important. Body needs glucose, at least some amount of glucose must be there. So at that time, a different hormone is produced, which converts glycogen to glucose back. And all these hormones are collectively called as the endocrine system. Let's have some examples of the endocrine system. So first one is the pituitary gland, which is the master gland, you can see here. Then we have a thyroid gland. Then we have pancreas. Then we have above the kidney, there's adrenal gland. There's a testis, and this is the ovary in female. Transport around the body. So endocrine glands have a good blood supply, right? It's very important because if blood supply is not good, then how the information will travel? So when they make hormone, they need to get them into the bloodstream immediately, as soon as possible, so that they can travel around the body to the target organ, and the target organ is going to respond. Hormones only affect the cells with target receptor, that is the hormone can bind to. They are either found on the cell membrane or inside the cell, so that depends. The liver regulates level of hormone in the blood, transforming or breaking down the new cells which are present in excess. So you can see here in this diagram, so brain has released some of the chemicals to tell pancreas to release enzyme called insulin, right? So you see they are traveling and they're going to the cells, and the correct cell will take that particular information. Important hormones in the body and what they do, what is the role and the effect. First example is adrenal gland. Source is adrenal gland, and the hormone is adrenal or adrenaline. Role: reduce the body for unusual condition like in fight condition or even in flight condition. And how they affect, they increase the heart rate and breathing rate so that more oxygen is taken by the body so that they can respire and provide continuous energy. Insulin secreted by pancreas. Role is to lower blood glucose level, right? So how they do? They convert extra glucose into glycogen. Testosterone present in testes. What is the role? Main sex hormone in the male, right? They develop secondary sexual character. Estrogen in female, which brings again secondary sexual character in female, and also begins menstrual cycle. Let's talk about glucagon. So glucagon is a hormone which controls the negative feedback. When we say negative feedback, The meaning is bringing something to the set defined value. And we have two hormone produced by the organ for negative feedback. First is insulin and the glucagon. Both hormones which controls blood sugar level are made in the pancreas. So that's very important. Insulin is produced when blood glucose level rise and stimulate liver and muscles to convert extra glucose into glycogen. So at that time, insulin is going to bind to the blood. Glucagon is produced when blood sugar falls. So it's the reverse reaction is performed by glucagon. So let's see here. Let's try to understand this whole process from here, from the left side. So normal glucose level, blood glucose level rises after a meal, right? So who will detect the increase in the sugar molecule in the blood? Pancreas. They will produce insulin. Insulin is going to stimulate the liver and muscles to take the food and convert into glycogen. In this way, blood sugar will decrease and normal set value will achieved. Let's see when the blood sugar is going to fall. At that time, pancreas is going to detect and going to release hormone called glucagon. Glucagon, what they do, they stimulate liver cells to break down the glycogen into glucose, and then this glucose is released into the body. And in this way, your blood sugar will come to normal or it will increase up to certain limit, right? So adrenaline, known as fight or flight hormone, produced in situations when the body may be in danger, right? That is either fight condition or flight condition. It increases a range of different things to happen in the body and designed to prepare for it. Like they are making ready actually, which includes increasing blood glucose level for increased respiration in the muscle cells because body is doing respiration and energy is going to produce. This will increase the pulse rate, breathing rate, so glucose and oxygen can be delivered to the heart muscle cells immediately. And at the same time, CO2 is taken away, diverting blood flow towards the muscles and allow non-essential part of the body such as your guts, your kidney, right? They are not going to get the blood. They will get, but it will be more to the upper part of the body. Dilating pupil to allow as much light as possible to reach to the retina so that more information can be sent to the brain and brain will give appropriate response. Apart from this is a very common example. Apart from this, what adrenaline do? Increase the concentration of glucose and also increase the heart rate. When it comes to homeostasis, right? Homeostasis means how we are maintaining a constant internal condition, which is very vital for the organism to stay healthy. There are a few examples here in which homeostasis is maintained: body temperature, water level in the body, and concentration of sugar in the blood. So let's talk about the role of insulin in the homeostasis. So insulin is secreted when the blood sugar is high. This is directly after a meal, immediately after doing lunch or dinner. The kidney can only cope up with certain level of sugar molecule in the blood. If the level goes too high, sometimes glucose is excreted or it is lost in the urine, right? So this is like running a car with a hole in the petrol tank. Valuable fuels is being wasted, right? To avoid this, insulin temporarily converts excess glucose into glycogen in the liver and muscles both. So insulin decreases the blood sugar concentration and the glycogen is converted back to the glucose several hours later when blood sugar dipped due to respiration in all tissues, right? What is negative feedback? When we say negative feedback, the meaning is bringing any condition to the ideal situation or to the set point or return to the normal condition as it was earlier. So how it works? So if the level of something rises, control system are switch on and it is reduced. right? In another way, if the level is something false, control system is going to switch on and it will raise it again. So negative feedback mechanism are usually a continuous and conditional cycle, which brings down and bring up so that overall they stay in a defined range. So like this, condition in the body changes from set value. There is a hypothalamus who will detect the change, correct the mechanism to come back to the normal. Condition reached to the set value. Corrective mechanism switch off. But again, the situation will come where something is going to disturb and we have to bring back to the normal. Blood glucose control. Blood glucose level are controlled by the negative feedback, and we have a two hormone which one is going to convert, another is going to convert back to the glucose, right? So both hormones are produced by pancreas, in which first is insulin. What they do, they produce when blood glucose is higher, and they stimulate liver cells and muscle cells to convert excess glucose into glycogen. Glucagon is produced when the blood sugar falls, and it stimulates liver and muscle cells to convert and store glycogen into glucose. And then later on it is released in the blood so that your body cells will get it. So this is the diagram which shows how the blood sugar is maintained. So let's try to understand this whole thing using an arrow. So let's see the red arrow. High blood glucose, right? High blood glucose means immediately after meal. So high blood sugar means it will promote insulin secretion. Pancreas is going to secrete that hormone insulin. Insulin stimulates the glucose uptake from blood, goes to tissues, and blood sugar will get lower. Or insulin activate the liver cell to convert all the glycogen into glycogen into glucose, and then lowers the blood sugar. When we have a low blood sugar, let's see this arrow, the green arrow, it promotes glucagon produced by the pancreas. Glucagon will stimulate glycogen to break it down, and then blood sugar will raise up. Now let's talk about the diabetes. Diabetes is a condition where the blood sugar level is not able to be regulated or maintained in a set value because insulin secreting cells in the pancreas are not able to produce the insulin. This means that blood glucose levels are often far too high, and it can be treated by injecting insulin from outside, but it's not a permanent solution. The extra insulin causes liver to convert glucose into glycogen, which reduces the blood glucose level, right? The basic symptom you will find in the person who is diabetic, that extreme thirst. They drink water a lot. Weakness and tiredness will be there because of the more sugar molecules, blur vision, weight loss, and loss of consciousness in extreme cases. People with type 1 diabetes have a monitor their blood sugar level throughout the life or sometimes throughout the day also, right? In the level of physical activity and their diet immediately after the insulin needed. They can also help to control their blood glucose level by being careful with the diet. So if you have a healthy lifestyle, you are exercising, of course your blood sugar will come down. Homeostasis, how temperature is being controlled. So control of the body temperature is called homeostatic mechanism, which is maintained of a constant internal environment. And this means that internal condition within your body, like temperature, blood pressure, water concentration, glucose concentration, need to be in the set value. If it is going beyond, then it will cause a severe problem. So in order to ensure that reaction is in the cell body can function and therefore organism a whole can live. Human body maintains the temperature, that is the 37 degrees Celsius, and this temperature is the best temperature on which enzyme works. If the temperature changes Beyond 37, enzyme get denatured. Now they are not active. They are not active to do the work. Structure of the skin. So let's see this. This is this structure they generally ask in Cambridge exams. So this is the hair, and this blue color is your free nerves connected to the body, to the skin. This is epidermis. Then we have a dermis layer. Then we have sensory neuron layer and fat layer, the yellow one. On the left side, you can see there is a sweat pore. These are the muscles which is connected to the follicles, your hair follicles, then capillaries, sweat gland, arteriole and muscle cells. So how temperature is being regulated by the skin? So temperature is controlled by the brain, and they contain some kind of receptor which is sensitive to the change in the temperature of the blood actually. So the skin also has a temperature receptor and sends nervous impulse to the brain via sensory neuron. And the brain is going to respond to this information by sending nerve impulse to effector in the skin to maintain the temperature within a narrow range of value, right? Maybe between 30 to 40. Fat tissues under the dermis layer, they act as an insulator and prevent too much body heat to come out from the skin, right? So let's understand this with using this particular graph flow chart. Normal body temperature, that is 37, increase in body temperature. Let's see, temperature has increased. Thermoreceptor, where in the hypothalamus will detect the change in the temperature. Increase the sweating. There will be a vasodilation, and here lies will flat against the skin so that the body temperature will decrease and the normal temperature is achieved, right? Another condition, when the decrease in the body temperature is slowly decreasing. Who will check this? Hypothalamus. What they will do? They will cause skin hair to erect. There will be shivering and vasoconstriction, and then slowly body temperature will come back to the normal. So responses to the change in the temperature, this slide is very important. So when we are hot, sweat is secreted by the sweat glands and they're going to cool skin by evaporation. So heat energy from the body is lost as liquid water in sweat becomes water vapor, a stable change. But in case of when we are cold, of course there will be a change. Skeletal muscles will contract, you will shiver. These are involuntary muscle contraction, need energy from the body, right? So that heat energy is produced. What about the hair? So hair lies flat against the skin, allowing air to freely circulate. This increases heat transfer to the environment by radiation. Erect hairs trap a layer of air around the skin, which act as an insulator, and it's going to prevent the loss of heat. Vasoconstriction and vasodilation. So as the name suggests, constriction, that is going to get constrict. Dilation, it's going to get dilated. Vaso means the blood vessels. So when we are cold, blood flow in capillaries slows down because arteriole leading to the skin capillary are narrowed. So that is why we call them vasoconstriction. And when there's a vasoconstriction, there will be reduced in the amount of heat loss by radiation. And when we are a hot, blood flow in the capillary increases because blood vessels to the skin capillary get wider. That's called vasodilation. This cools the body as blood is flowing as faster at rate through the skin surface so that more heat is generated and the body will be come back to the normal. So see this, these are the hair follicles connected to the muscles. These are the blood vessels. Response in the skin when cold. So you can see. So air is being trapped here so that once air is trapped, that means temperature will slightly be better. Let's talk about the tropism. Tropism means the movement of plant in some different conditions. So in this topic, we'll talk about gravitropism, also called geotropism and phototropism. So plant can respond both change in environment, that is either light, water, or earth gravity. Their response are usually much slower. It's not a fast process, right? Because the hormones need to be transported and then organ have to recognize these messages. So it takes some time, right? So they grow either towards the stimulus, that's positive response, or sometimes away from the stimulus, that is negative response. And these collectively responses are known as tropism. And it's very important to a plant that is root and shoot grow in the right direction. So shoot must grow upward, root must grow downward. That is very important. So this means that shoot have a positive geotropism response and negative geotropism response too. How? So shoot at the same time there is one thing is going on. Root need to grow downward into the soil, away from the light and towards gravity in order to anchor the plant and absorb water and minerals for a longer time. This means that root have a negative phototropism mechanism and positive gravitropism, right? So these responses are very important. So gravity response is gravitropism, sometimes called geotropism. Definition growing away from the gravity. So it's a positive. Negative grow away from the gravity. Light phototropism growth towards or away from the direction of the light, right? So if they are going towards the light, positive. If they're going away from the light, negative geotropism. Sorry, negative phototropism. This is a very common experiment we all have done in our life. I have literally done this. Sometimes I keep the pot like this, and after a few days, we'll find that the shoot is going in upward direction and the root is going in downward direction. There is a small investigation has been done to check this. So these are the setups. So setup, this is the box, light proof box, and one plant is kept in this one hole, and through this hole unilateral light is being passed. In this setup B, the clinostat is rotating slowly, and in setup C everything is closed. It's a closed container. So the seedling in A grow towards the light source, right? In B the effect of the light only coming from one direction and has been cancelled out by using clinostat because they are moving. This means all sides of the seedling get an equal amount of light, so they are not going to curve. But in C, it's looking for light and the plant becomes tall and slender with yellowish leaf due to lack of sunlight. So we have seen in the third, its container is packed. If we have to investigate geotropism, it's very easy. You can put the seeds in the cultivar and put it in the petri dish. So there's a setup A and there's a setup B. So let's talk about setups. Add some damp cloth wool into the wet petri dish. Place 3 seedlings. Control hole in each petri dish. So A, B, C. We have seen the diagram. Cover each dish with a lid. Attach one petri dish to the support. Attach a second petri dish in a clinoset so that we can maintain the movement, both placed in the light-proof box. After 48 hours. Wow. So result is all radicals have grown downward. You can see that is positive geotropism, and all pumil. Pumil is this one, the green color. They are going up. That is negative geotropism, right? Investigating the gravitropism response. So in the first petri dish, all radicals have grown downward. That means it's a positive geotropism, regardless of which way they will initially facing, horizontally, vertically, or up, down, whatever. And all pupils have grown upward. That is negative geotropism. In the second petri dish, all radicals at the pumil have all grown either up nor down, but straight outward. in whichever direction they are placed, as the effect of gravity has been cancelled out by revolving nature of the clean stack, right? So they have shown no gravitropism at all. So the experiment need to be done in the light-proof box in order to cancel out the effect of light on the growth of the seedlings. Auxin is a chemical substance and they control the growth, right? And this is a very basic plant hormone. It's a growth hormone in plant which controls the direction of growth of root and shoot. So therefore we see plant control their growth chemically actually because they are producing hormone. Oxygen is mostly made in the tip of the growing stem and the root and can diffuse to the other part of the stem or root. So spreading from one higher region to the shoot tip down to the shoot as an area of lower concentration. So the food will also transport. Oxygen stimulates the cells behind the tip to elongate. So the more oxygen there, that means faster they are going to elongate. If sunshine at all the tip, oxygen is distributed evenly throughout and the cells in the meristem grow at same rate. So yes, oxygen requires sunlight, right? So when light shines on the shoot predominantly from one side through, the oxygen produced in the tip concentration on the side, shaded side, making the cell on that side elongate and grow faster. That is why there is unequal growth on either side of the shoot, causing shoot to blend and grow in the direction of the light. Like this you can see. So oxygen made in the tip and sunlight is fall here. So they are going in opposite direction. And wherever the oxygen will go, elongation will help in bending. Again, let's talk about this. So set A, B and C. So A, B is covered, C is moving, right? So hope you have the idea why C is moving. Investigating the phototropism results. So let's talk about this. So treatment, they are seedling A, B and C. The tip of the stem have been removed. No light reaches the tip and more light reaches one of the tip. Here you can see ABC. Effect of oxygen, no oxygen produced in seedling A. Equal concentration of oxygen both sides of the tip. Greater concentration of oxygen on both sides of tip. Results, stem do not grow longer. Of course here stem is going to grow and here also. The reason this provides that oxygen is made in the tip and is needed to stimulate cells elongation and therefore growth without the tip, and therefore without oxygen there is no further growth in stem. In seedling B it proves that if the effect of light or oxygen that causes phototropism, there is no uneven distribution of oxygen. But in seedling 3, the C1, there is uneven distribution and oxygen is helping shoot two bends towards the light as seen in the diagram. So what we have understood that oxygen play a role in plant response to gravity, affecting plant root and shoot in different way. When shoot grow away from the gravity known as negative geotropism, gravity modifies the distribution of oxygen so that accumulation of the lower side of the shoot. As seen in the phototropic response, oxygen increase the rate of the growth, either it shoot or root. When root grow downward towards the gravity, known as positive geotropism, so in root, higher concentration of oxygen results in lower rate of cell elongation, right? And oxygens that accumulate at the lower side of the root inhibit cell elongation. So as a result, lower side grow at a slower rate. And this causes root to bend downward only in one direction. And that's it. Thank you for being a patient listener, and we'll catch you in the next uh, video. So far we will be in touch. and don't forget to um, work on my videos, check the latest videos, and work on all the videos and give your responses, and I will be happy to help you out. Thank you, and that's it.